It's my great pleasure to present our next keynote speaker, Michael Breakspear. I have to say this makes me happy for several reasons. One, because it acknowledges on the part of OHBM the emerging field of neural modeling, which started in the 70s but only now has hit full stride, and a lot of that's due to the work of Michael himself. Second, because this is an acknowledgement of the work of Michael, who is one of these rare individuals who is not happy with being confined within a box of one specialty. He's a full psychiatrist, he still sees patients, and on the odd hours, he writes down equations. And what he's extremely good at is at making people understand what these equations mean and turning them to the benefit of the understanding of the brain and its disorders. He did his medical studies at the University of Sydney. When you hear him talk, you'll know he's basically an Australian guy. He finished in 1994. He did his postgraduate studies in psychological medicine at the University of Sydney, finishing in 2003. His postdoc until 2009 was with Peter Robinson, who was also one of the pioneers of this type of modeling. And he's a fellow of the College of Psychiatrists and currently group leader of the Queensland Institute of Medical Research. Just to finish, because what we want to do is actually hear Michael, I have to say that the field of neural modeling, which started out in the 70s with very simple dots and points of neural masses, has actually recognized that the brain is a sheet. It's an extended spatial surface which allows dynamics to operate as waves. So it's not surprising that Michael loves waves. In fact, when I stayed with him a few years ago at his house, every morning he went out to surf. You can see how interested he is in waves. And precisely he's going to talk to us about this field in which he has done research, which is neural field theory, and he's combined it with studies in connectomics. And as I repeat, he is both an excellent medical doctor, but also an excellent researcher, and even more, an excellent advisor and friend. So, Michael.
Thank you very much, Pedro. And it's a great pleasure to be introduced by a Cuban neuroscientist during the big thaw. And great to see Pedro's pioneering work highlighted last month in Science and PNAS. So hats off to the Latin American chapter. Okay, it's a great pleasure to be here, and thank you all for coming. I hope I can do some justice to what Pedro said about making maths understandable to a broader audience. I'd really like to thank the organisers for inviting me. It is a great pleasure indeed to be here, and to use some local vernacular, it's a really banging conference so far. Okay, so on to the talk. I'm going to talk about brainwaves, and this is work, most of which hasn't been published yet, and James, Leonardo, and Alistair, who's a very bright student, but James and Leonardo are here. So what will it all mean? Okay, so I did physics at university, and I stumbled as a young man into the library and grabbed some books and thought if I really concentrated hard enough, as an arrogant young man, I'd understand it. But it actually took me a long time. But I always really loved these great universal theories of things, and distilling the essence into a set of equations that explained and harmonised things. That's what I'm trying to do with my work. And if we look at these great theories of the atom, model of the universe, and a model of electricity, some of them are very simple, E equals mc squared, and some of them more complex, like Maxwell's equations, but if you study them, you can understand them. But what is the model of the brain? And this is a question, of course, that hasn't been resolved and may never be. But there's certainly opinions, and some would say a model of the brain, if you really want to understand the brain, you need to understand the full brain, and the best model of the brain is therefore the brain. And some might even say preferably the same brain. Some might say you've got to understand neurons, and if you understand the neural code, get down to that level. And of course, in our field, we've been swept along by connectomics and the bid to understand this. So it's a broad church, and I'm not claiming any sort of salience, but the work I've been working on with many others, and Pedro mentioned some of the pioneers, is to posit an evolution equation. So this is an equation that doesn't describe the motion of the planets or the field of a proton, but the evolution of states in the brain. And by states, I mean things like firing rates, conductance, channel capacitance. So these are things that are very hard to measure, but this is what is actually happening at the level of the brain. And these evolution equations typically encompass two properties: a local circuit equation, which I've caricatured like this, and then some other component that introduces coupling from afar. And these come in various sources. Now, this is the huge conceptual jump for systems neuroscience. We're not talking about the states of individual neurons, but as I'll revisit in a few minutes, we're talking about population densities. So these are the states of hundreds to thousands of neurons, and that's the appropriate level of description that I would contend for human brain mapping, because this is the sort of force of neural activity that leads to the signals that we measure. I've put some of our work here, but many other people have worked in the field. So these are called mean field or neural mass models, and we don't measure those, and so it's very important, just as Newton described optics before we could understand the motion of the planets, that we have an observation equation. We've worked hard on observation equations for the bold, and this is work by Kevin Aquino and me and Peter, looking at unfolding bold across the cortical sheet. And so X becomes Y, which is what we measure. And these are sort of causal models. We can have debates about the notion of causality in the brain, but they're causal in a sense is they're models of what we think causes the data that we measure. And of course, Carl Friston pioneered this approach through DCM. Okay, so what am I going to do in the next 30-odd minutes? I'm going to just dissect this evolution equation a little bit more deeply. And then I'm going to do a case study, and this is work that we've been sweating away in Brisbane for the last few months of a particular application, because I think that's instructive. And very briefly, I'll try and wrap up what it actually might mean for how we understand the brain. Okay, so here's the brain. We're not going to study the brain, we're going to study abstractions of the brain. So we tease apart many of the details and we come up with equations that embody some level of abstraction. And then we can do formal analysis with maths, particularly stability analysis, or we can throw the code into MATLAB or Python, and we can do simulations. Both are important. Analysis can tell you some very deep results. Simulations can tell you about phenomena that you can't fully understand with closed sets of equations. These can be detailed neurophysiological models. This is a cartoon of a corticothalamic neural field model after the pioneers of people like Walter Freeman, Paul Nunez, Lopes da Silva. In the 1990s, it went to a new level with work by Vic Yersa, Herman Harkin, Peter Robinson, and this is Peter's corticothalamic model, which I've spent quite a bit of time doing. These incorporate dendritic filtering, axonal delays, cortical anatomy, lots of beautiful anatomy. But sometimes we might just want a very simple algebraic form, and I'll explain why. And so we can also use very simple canonical models. And with maths, we can reduce the former to the latter. So these are connected. We're not talking about a zoo of models. We're talking about a small number of animals, hopefully. Okay, so we do the analysis and simulation. We put in our forward model. We get a prediction. We take the brain, put it in a scanner, and we get an observation. So we can then compare our model predictions with our observations. If we change the forward model from the hemodynamic model to Maxwell's equations, then we're predicting the lead field or EEG. So we're using the same generative models, hopefully. We don't want a different model for everything that we measure, but now we're explaining a different phenomenon. If we use these simple algebraic equations, then we can use variational methods and invert those models to recover the likelihood of their relative likelihood, and parameter estimates. And this is also important. Okay, so that's the general framework in which we work. That's what we wake up to in the morning when the sun comes up. And I'll just try and illustrate this now with two related case studies. The first study will be a study of the human connectome, but with a particular computational bent, or physics bent. And then I will demonstrate the use of neural field or neural mass models to understand aspects of the connectome. Okay, now this work was just published in Neuroimage. It was done by a very bright PhD student, Alastair Perry. We did what a lot of people do, and I know this is, of course, not a closed field yet. We took diffusion-weighted imaging. We deconvolved that, being from this background of forward and inverse models with the constrained spherical deconvolution of M-matrix. And using probabilistic tractography, we then created a brain of fibres, or I should say streamlines, of course. It's an inference that's imperfect to call them fibres. We use Andrew Zalewski's parcellation to get regions that are approximately similar volume. This is important. And we get a connectome. So if we turn these parcels into nodes and the streamlines into edges, then we have this network representation that, of course, Olaf Sporns spoke about 2 years ago. And just to note, because I'll be showing these matrices, the edges can be along the sides of a matrix, and the nodes along the side, and the edges will be these little colours. Okay, a bit of a sanity check, and I hope you don't see streamlines that you don't think should be there. If we do a contrast using NBS of left greater than right, we get the perisylvian fibres in the arcuate fasciculus. That makes sense, right? That's where the language cortex is. And if we do a contrast of right greater than left, then we get the ventral temporal projections to the visual field and inferior longitudinal fasciculus, which I'm told the right hemisphere is somewhat specialised for these rapid visuospatial things. And I hope you appreciate these projections from 3D to 2D look like some of the fibres are going through ventricles, but Alastair spent a lot of time correcting that. That's just an artefact of the projection. Okay, so here's our network. Following the wonderful work of Martin van der Heuvel and Olaf Sporns, we dove in and looked at the Rich Club, and these are older healthy Australians. So the Rich Club are this constellation of high-degree nodes. This is structural data, not functional data, but they've been described in both. And I'll revisit this in a minute. The feeders are the next hierarchy down. They're nodes of lesser degree, but they connect to the Rich Club. Actually, if you do social network theory, these are the guys these are the dudes that you want to influence if you want to influence the whole system. The Rich Clubs are already sort of slave to the network. And then we've got the peripheral nodes out in the periphery. Now, what's important here, and I know I can't show all three, is the hub-to-hub connections are longer than the feeder or local connections. And this combination of dense, short-range local connections causes clustering. We'll come back to that. And these rich, long-range connections give our connectomes the so-called small world topology. It's not magic, it's just this aspect of anatomy. And this constitutes the topological centre of the brain. So that's the first definition, and I'm going to talk about the geometric centre in a moment. But this constellation of high-degree topological hubs. Using the brain connectivity toolbox and a modularity algorithm, we were able to, Alastair was able to recapitulate what's been in the community by people like Danny Bassett, and looking at these modules, 6 or 7 of them. What you see here are the two hemispheres. Sorry, I can only point here. And you see this frontal and precuneus midline areas straddling both hemispheres, whereas the visual cortex doesn't. That also is consistent with my anatomy, which is not as deep as some in the audience. Okay, so there's the topological representation of the connectome. Of course, we can look at other measures, but the point I want to make, well, it's been made by Danny Bassett and Peter Robinson and James Henderson, is we've kind of disembodied the brain in this network approach. And the brain is spatially embedded, so we should go back in the other direction as well. So we have this lovely tractography where we wanted to test some of the predictions that had been made. How do we do that? Okay, just before we do that, so purely topological networks of things in which you can have long edges that have no more cost than short edges, like Wikipedia, scientific collaborations. Pedro and I love working together. He's in Cuba, I'm in Australia, but that doesn't matter because there's no cost there. I'm more likely to work with Pedro than someone sitting in my own institute. I didn't mean that to be funny, but it's true. Spatially embedded networks are those in which there is a cost of edge length. And these are things like brains, highways, and cities. That's why we have cities, because it doesn't make sense for people to go, at least before we became an e-world, to go a long way. People wanted to go from office to office and have coffee together. So cities are great, spatially embedded, complex, dynamic networks. Now, in theory, if you're a spatially embedded network, and you've got two neighbours that are near to you, they'll also be near to each other through spatial assortativity, and then therefore they're probably connected. So already you're going to have clustering. This is the idea. Okay, this is the effect of distance on our connectome. So we've got the connectivity matrix in the middle and the interareal distance on the right. And you can see what's red in one is blue in the other. In other words, they're inversely related. And importantly here, you can see the hemispheres, right? Left hemisphere, right hemisphere, interhemispheric connections looking a lot weaker, but also being a lot further. So this is edge weight or normalized streamline count, and this is distance. And we haven't made these sparse yet. We've got that happening in another study. If we plot this, you see most edges are very short. This is 2 centimetres here. This is 3 here. This has gone down by an order of magnitude. So this is the spatial embedding of human tractography. If we take log, because we don't want to just look at this, it's hard to see any structure, we take the logarithm of this axis, we get something that looks like this hairy guy. Now what's interesting here are a few things. First of all, the homologous left-right areas, which are the red, tend to be above this mean line. But interestingly, the intra and interhemispheric connections more or less follow the same form. So they're only weaker because they're further. This is slower than exponential, and we get a few little bumps up here. Now, if we look at these bumps, and James did this painstakingly, we see these are long-range connections. These are particularities of the data that are edges that are stronger than expected by virtue of their distance. If we just thresholded brute force, and this is what we're trying to get rid of, and James has a poster on this, we lose these sorts of connections. So spatial embedding is important. Now, if we want to benchmark the network properties of our real brain with a certain number of nodes and edges, we need a reference network. And many of you, or all of you working in the field, will know that we use random networks. Now, they're not spatially embedded, so we're not comparing apples with apples. So we're going to make spatially embedded networks, and I'll tell you why. I'll tell you how now. We fit a bit of I'll try and go through these not too quickly. We fit a low order, here it's just I think it's a polynomial, third order to the mean. Actually, if you turn this into streamline distance and not Euclidean distance, this is almost perfectly linear in these linear logarithmic coordinates. We regress that out, that's easy, right? We just subtract it. But what are we left with is that there's also some spatial embedding of the variance. These short-range connections are pretty anisotropic, similar everywhere. This is the connectome, really hiding down here. This is this variability, and we want to preserve that. So we can do that more or less to second order, and that's what the edges look like. We've got something that looks like a Gaussian, which means those of you who have done resampling, and Tom Nichols is here, we've whitened the data. And that means we can shuffle it like a pack of cards, or we can draw new edges from this Gaussian distribution. Then we can throw them back in, and then we can put the geometry back just by inverting those other two steps. Okay, so these have to second or third order the same spatial embedding. This has got topology, this has got no topology. The topology is gone. We shuffled it in the space where the data was white. Okay, now we have to play a little bit more algebra to get these high degree nodes back. We can do that. Here they are. That's the original. The blue is, sorry, that's the distribution of log node strength. We want hubs, so we have to play around a little bit to put them back in. And we put them back in the brain. Right, so it's now got the actual geometry of the brain. Here's our original connect home, and here's our surrogate geometry preserving network. We've whitened the data a little bit. Don't get too worried about that. It looks pretty good, right? But already we see these little ears have gone. And that's important. And the red interhemispheric homologous connections are down in the cloud now. It's just one big cloud. These are particularities of the real data to topological features that we've randomized away. But we haven't done this. This is a random shuffling algorithm, and the same with the Sneppen algorithm. We haven't destroyed the geometry. Now, if we take our modularity decomposition and apply it to a random geometry destroying network, we get this modularity is plummeted from 0.6 to 0.19. Now, this was, I guess, predicted by James and Peter. What happens to our geometry preserving? Now, there's no topology here. This is just inherited from the geometry. And lo and behold, there's a whole lot of community clustering. And here's the two hemispheres, but there's nothing in the middle of the hemisphere. So a lot of this topological specificity is gone, and some of it's come from geometry. We can do it with the clustering. Here's these local feeders all close to each other. This is highly clustered compared to our random network. And here's this geometry preserving. So much of the modularity, nearly all of the modularity, and a large component of the clustering comes from spatial embedding. I have to thank Steve Smith for asking me to write a paper, and this is when we got into this a little bit. What about the rich club? Okay, here's our topological centre. So is the geometric centre of the brain the same as the topological centre? Christoph? I'm joking. No, it's not. He's always asking questions. Not surprisingly, the rich hubs informed only by geometry in the middle of the brain. This is what James and Peter predicted, and here they are, but it's not like the real brain. There's a mismatch, which is pretty cool. This is what the mismatch looks like. The red guys are those nodes that are richer than they would be, and the blue guys are those that are poorer. I mean guys in a non-gender specific way here, by the way. And you see that the rich club has been pushed through some sort of pseudo-evolutionary force out from the centre of the brain into the periphery, the occipital cortex, the inferior frontal gyrus, the anterior insula, and the prefrontal poles. What's really cool is the anterior insula, which is my favourite part of the brain, is the only region that's both in the topological and geometric centre. And maybe that's because it also needs to be close to brainstem centres for its role in interoception. Okay? Now, here's the rich club of the real brain. There it is there. This is richness relative to random networks. There's the hub. And this is the rich club of the geometry-preserving brain. And it's, look at this axis, 1.05, it's way richer. So in moving away from the geometric centre, the brain has preserved its richness, but not completely. In fact, if you look closely, and this is also in Martin and Olaf's lovely work, it's cartel-like at high degree. In the real brain, these high-degree hubs have actually moved away from each other, like bikie gangs having their own turf. In Queensland, we had anti-bikie gang laws, so we don't have bikie cartels anymore. Okay, so it's not like a city, it's more like two big cities, which is interesting because, once again, social network theory or economic theory will tell us that duopolies are not very stable. We have Coles and Woolworths in Australia. So what does that all mean? Okay, to understand what that means, I think I've still got some time, we're going to use neural mass modelling. We kind of have to stop there just with anatomy. So we're going to use a neural mass. There's our circuit. It's a pretty simple model. Pyramidal cells, inhibitory cells, and the pyramidal cells have a potassium conductance. That's called a Morris-Lacar cell. But then there's population determinants of firing rate, and the inhibitory cells are slow. Yada, yada, yada. Looks better like this. We can plot the time series, V, W, and Z, fast and slow. When these burst, the inhibitory cell builds up and turns the bursting off, and then the inhibitory cells don't get any juice, so they go down. This is like up and down states. If we change the excitability, though, we go through some bifurcations, and we get this guy. It's called a homoclinic chaotic attractor. Still the same equations, just one parameter has been tweaked. I made quite a lot of papers out of this little chaotic attractor. Okay, so there's our local network. Now we put in a whole lot of these in the different cortical column, and we couple them together or to channel Guzdeco. We let them feel each other through the connectome and see what happens. Now in the movies I'll show, the state of each node is represented in colour, right? Now we worked on this, Chris Honey, Olaf Spons and Rolf Cotter, but also Guzdeco, Vig Yersa, Petra Ritter, and my group with Leonardo Goyo have also been working hard on this area. Okay, let's see what happens. Is it just going to be complete chaos? The answer is no. Through the magic of nonlinear dynamics, this complex system organises after a few seconds from random initial conditions into these beautiful geometric forms, rotating waves, breathers. I didn't make these terms up. Mathematicians have been quietly studying them in their hallowed hallways for many decades, and travelling waves. These come quite naturally in excitable media with these sort of local spatial kernels. Now, these are all the same equations, all the same parameters, completely stationarity at the level of the evolution equation. Do they just go on like this forever? Okay, this is where I have to be a bit patient. Here we are. This is one particular pattern going over to the right hemisphere, and now the left hemisphere starts to develop some symmetry. It slows down, loses a bit of order, and now it's gone into a different regime. If we watch it a little bit, now it's kind of rotating, stays like that. Something happens, and now we get an eye up in the top right. Okay, so back in Brisbane, I said to James Roberts, get me an interesting ohmmeter out of these dynamics, and he did. We looked at the cross-correlation between phase dynamics in the two hemispheres. This is a very crude thing, but it's much like at the neuronal level what Marcus Raichl is talking about through his lag thread networks that was recently published in PNAS. When these are these patterns, this is the time delay, this is the time, and you see these transitions. Just looking for periods of low phase synchrony, we can partition this. So we've gone from waves to formal metrics. Then we can say, well, okay, this is a neural state equation. We can use this to predict. There's time delays in these connections too, I should say. We can convolve with Kevin's hemodynamic response function because the nodes are quite close. We can get a prediction. And this is actually work I did with Andrew Zaleski and Alex Finito, who are here. Andrew uploaded the Human Connectome project, looked at sliding window functional connectivity, and found this striking non-stationarity. Now, it's important that the equations are stationary. The statistics of the observations are non-stationary, and I think that nuance is a bit lost in the field at the moment. We can change the forward model, and I'm sorry if someone in the audience did this. I just took it from Google. And we can project it into the lead field, and we get something that looks like a paper that Tjerd Boonstra and Said Makanoun published recently, and these brain modes in channel space. Now, as Pedro says, we have to do the network at the source level, but we get an image in source space. And these are like the microstates of Christoph Michel. Okay, this is now a metric we can use to compare the brain as it is to the brain as it would be if everything was connected according to geometry. These are a whole lot of simulations, all partitioned. There's 25 there's 5 long ones. These are these metastable transitions. I can use that word hand on heart because I know these dynamics are following something called a heteroclinic cycle. A guy called Carlo Lang has studied that using a lot of beautiful maths. And here's the geometric null. Already, you see, I don't think I can go backwards, but I'll show you a comparison in a minute anyway. Oh, there's a little red button. Aha. The stability is deeper and longer, and the metastability is weaker in the real brain. We can then quantify that, and you can see, oops, daisy. Multitasking. Oh, God. By randomizing edges between no randomization, in other words, the real brain, 10, 20, 30, up to 100%, we can slowly go through this transition from geometry to geometry plus topology, and we can just measure the phase synchrony between the two, phase lag synchrony between the two hemispheres and get a number, and the error bars come from repeated simulations, which Leonardo Goyo ran till late at night. And here we go. Greater stability, greater metastability in the brain. So something happens here. Leo and James have looked at something called percolation theory and seen a lovely phase transition here. But that's another story. Okay, what does it mean? In summary, in the first study, computational modelling showed that the rich club is actually not as rich and certainly not as geometrically central as it would be if determined by geometry alone. Using neural field or neural mass modelling, I should say, we showed that this loss of this cartel-like property of the brain endows the dynamics with greater metastability. What does it mean? Okay, here we have to pontificate and be speculative. The brain is less stable, more metastable, because structural hubs have a subtle cartel-like topology. We kind of know this because we get rid of the cartels through the randomization. Now here's Carl, I'll channel him. Metastability might be crucial for perceptual and motor ignition. Actually, the notion of perceptual ignition comes from Stanislav Dehaene, that this is what attention does to sensory inputs to cause conscious perception. Work that I've done with Vin Nguyen, Ross Cunnington lately, and you would have seen Ross's talk yesterday, is that in voluntary motor pressing, if you look at EEG fMRI data, you get something in your models that looks like weak transverse stability. Too much system stability, according to this notion, would be maladaptive, and this could be dwelling on negative thoughts, such as in melancholia. And this is work that we've just recently published, actually. Matt Hyde and Christine Gao have looked at functional connectivity in Christine's case, and effective connectivity in Matthew's case in patients with melancholia. And phenomenologically, they get stuck for long periods of time in these really dysphoric states. So, you know, we're not just arm waving, we're actually inverting some of our data to estimate the parameters of these models using DCM. Okay, since I have a minute more, I'll segue here. How does this relate to cognition? Okay, so I'm going to make a bit of a conceptual jump here. These little mean firing rates are representing something, a value, either in sensory cortex, a value or a belief about the world. In higher level regions, maybe they're representing values that are encoded in lower level regions. So that's fine. That's what we think we do in the world. We represent things in our brain, the value, the brightness of the sky, the redness of something that's red. And that's all fine and dandy, but if we look in computational behavioural neuroscience, work by people like, Christoph and Tim Behrens and others, we need value, but we need the precision of those representations. And our means don't have variance. So to do this, we go to something called the Fokker-Planck equation. Do I have that there? Oh, no. Fokker-Planck is a model basically that encapsulates both the mean and the variance. And so this is like put in each region instead of the mean field. This is not done yet. This is a conceptual thing. Of course, I can easily make movies. So then each region sees not only the mean of each other region, but the variance. And that's bringing in the spiking a little bit more. You can do other approximations, expansions. And this is a lot like work that is being done in terms of Bayesian inference and active perception, where both the mean and the precision or the inverse variance of the world is represented. Okay, thank you very much. I'd just like to thank my group. You saw that James, Leo, and Alastair, who is back in Brisbane working on his PhD, but also Vin, Christine, Matt, Sasha, who's worked very hard getting our multimodal imaging laboratory up and running, so he doesn't get the glory at the moment, but that's a big part of what we do. Anton and Tjerd, and many thanks to my friends and collaborators and supervisors, and I'd love to name you all, but I'd probably miss one name and then I'd feel really guilty, so thank you very much. Okay, so we have a few minutes for questions, if there are any. Yes, over here.
Thank you. Thank you for your wonderful presentation. One of the things that you showed in the beginning is that the weights, how the weights of the anatomy, how they depend on distance. But my question is, and also you use this distribution to normalize random networks. My question is, should you not account for the fact that all the white matter fibers that they should maintain within the cortical layer? So is it correct to use the Euclidean distance, but should you not use constraint distances to get
actually, most of the work we've done is actually taking the median streamline length, not the actual Euclidean distance. And I just didn't put that in my slides, but it does make it more exponential. We know there's imperfections in probabilistic tractography, and all I can say is that those imperfections are propagated into our nulls. And of course there was a paper just last month, I think, in PNAS showing that tractography gets confused with all these U fibres, or these parallel fibres in the base of the gyri. But those errors are also in our reference networks. So I don't know if that completely answers your question, but of course tractography won't pick up, I understand, connections within the grey matter itself, because it's more or less isotropic diffusion there.
Okay.
Maybe you can ask me during the break if I can. Do you have another comment?
No, thank you.
Okay.
Hi. Thanks for the fantastic talk. So you were talking about the Rich Club, and then we're talking about, the sorority, feeder hubs, and the cartel-like structure, which seem to be like metaphors, but pretty fitting metaphors. And in philosophy of science, we know since decades that metaphors are very, very important in science. But I wonder whether you think that they are essential to make sense of the analyzed data that you were presenting. So in other words, can you reformulate the results about the Rich Club without using these metaphors, or do you think that they are absolutely essential?
I agree with you about the power of metaphors. I was always using, when my kids were young, metaphors from Finding Nemo to my lab group, and I think it's helpful. But you can go too far. Now, all these things have mathematical definitions, so we can write down the Rich Club as a mathematical equation, and that's the beautiful universal language. But unfortunately, there's an inverse relationship between the number of mathematical equations in your paper and its impact in biology.
Thanks.
But yeah, it's important to do that, otherwise you're arm waving, and that's the way to tie metaphors and true definitions together, I think. Thank you.
Thank you.
Maybe over here.
Hi, great. Great talk. Thank you very much. I'm wondering if you could speak to the selection of the parameters for your neural mass models and their relevance to physiology, both in humans versus animals versus disease states and things like that, and how the selection of those parameters changes your results.
Oh yeah. That cuts pretty deep, that question. Two answers are yes, it means a lot, and no, it doesn't really mean anything at all. It means a lot. It's the Achilles heels of simulations with complex models which are too hard to invert. Unless you take a complex model, and we've almost done that with Peter Robinson's corticothalamic model, and do a Taylor series expansion and get a simple DCM-like model which we can invert, then we've estimated the parameters which we can put into our fancy simulations. If we change, nonlinear systems are quite sensitive to parameters sometimes, so that also hurts. What's banking on our side is that the dynamics just depend on very simple things. Nonlinearities, exponential-like spatial kernels, global topology, and out they all come as spatial instabilities. Peter Robinson will speak about this tomorrow, I think, and it doesn't matter so much about the parameters. But that's why we also do quite a lot of formal maths. So both are helpful, I think, but you've got to make very careful metaphorical interpretations of your models. But we do, in some of the models, incorporate a lot of prior physiological knowledge. We can do this if we're inverting them using priors. And even in this neural mass model, of course, the parameters have physiological meaning, and they're meant to mean something. Slow fluctuations in inhibitory firing rates, faster fluctuations in pyramidal cells. And Elaine Testecchi showed some beautiful data that looks like these spatial kernels as well. So thank you.
Thank you.
So,
maybe one more question?
Yeah. Given the fact that the connectome based on tractography is full of false positives and false negatives, have you tried to do any of this on a system that is maybe better known, like tract tracing in monkeys and primates?
Yeah, okay, very good question. Just two answers. First of all, we're not really tractographers, but we're working to try and get rid of the false positives in probabilistic tractography by looking at inter-subject consistency, which is the same as deterministic algorithms people do. Yes, we've coupled these neural masses together with a compilation of macaque direct tracing studies, or COCO-MAC, that Klaus Stefan and Rolf Cotter developed. You don't get the local connectivity so well. Certainly if you look at it, and you only really get 1s, 2s and 3s, you don't get this beautiful continuous streamline count. There's definitely spatial embedding. We just uploaded some of the connectomic data, I think, from Science, was it so? There's mouse connectome. It's there, but I think there's a lot of false negatives locally in those data. You don't really see the synaptic footprint that you see in neurophysiological data, but you see more these long-range heterogeneous connections. But we can do that. That's what the Honey paper did, actually.
Thank you.
One more.
Yep.
To your left.
Oh,
To your left.
I'm a bit short-sighted.
Further
Okay.
left. So, so far as I see, what you showed us was an autonomous set of simulations, a very neat feeding of local circuitry into connectome. What are the prospects for extending this to show the impact and influence of sensory stimuli? So you showed lots of waves. I was thinking of migraines, but I guess I'd like to see consciousness.
Yeah, well, this is a model of spontaneous dynamics. What you can say is that if you perturb the system in one of the states of high synchrony, it's pretty robust to the perturbation. If it's in low synchrony in a transition, then you can really move it to a different trajectory. That's a little bit like this notion of perceptual ignition. But I think we're a long way from a harmonised theory. Unless you do stability analysis and treat the system as a very weakly damped system, and then of course you can look at perturbations and how they propagate. But you lose a lot of the excitability and metastability that we've actually measured most directly, I would say, in EEG data. Yeah, well, there's a lot of work to be done, but that's pretty good news for the young people.
Right, thanks.
Okay, thank you. Okay, thank you very much.